Hi there. Have you ever wondered how that swab you took from your nose during a COVID-19 test tells you whether you're positive or negative? The answer lies in an incredible, powerful technique called RT-qPCR. It stands for Reverse Transcription Quantitative Polymerase Chain Reaction. I know the name may sound very long and difficult, but don't worry, I'll break it down step by step so you can understand how scientists use this process to get the test results. First, we need to know what's in the sample. When you get a nasal or throat swab, the sample contains a mix of cells, mucus, and potentially viruses if you're infected. If the virus is present, it contains a genetic blueprint that allows the virus to make more copies of itself. Unlike human cells, which store genetic information in the DNA, viruses store their information in RNA. RNA and DNA are very similar, but RNA is single-stranded, and it works as a messenger. RNA's job is to tell the virus how to make the protein it needs to be able to function. Okay, so now that we know what's in the sample, we can learn how the scientists use the sample inside the lab. The first step in the lab is to extract the genetic material from the sample. They start with the sample, usually from a nose or throat swab, and treat it with a special chemical. These chemicals will break open the cell and release all its genetic material inside to the outside. It's kind of like popping like a balloon, so that everything inside will spill out. What we're especially looking for is the RNA of the virus. But at this point, everything is mixed together. So the human genetic material, the proteins, and even possibly the viral RNA. So the next step is to separate and purify the RNA, which is what we're interested in. Scientists will use tools like magnetic beads or filter columns to be able to extract the RNA and release everything else that we don't need. Okay, so the next step is reverse transcription. Here's where the RT in RT-qPCR stands for. RT stands for reverse transcription and it's an essential step. The PCR process only works with DNA, not RNA. But remember, as I previously said, the genetic material in the virus is inscribed into RNA. So we need to convert the RNA into DNA. To be able to do this, scientists will use a special tool called reverse transcriptase, which is an enzyme. This enzyme can be thought of as like a molecular translator, since it will read the viral RNA and translate it into DNA. This new strand that we made is called complementary DNA or cDNA. This step is critical because it gives us a stable DNA version of the virus genetic code. Once we have the cDNA, we're ready to move on to the next step, which is the PCR. So in the PCR process, we're gonna make millions of copies of a specific section of the DNA. Scientists would add short pieces of DNA, which are called primers. They were matched into a unique part of the COVID-19 virus. These primers are like a markers and they guide the enzyme called DNA polymerase to the target area. Then the sample will go into the machine, which is called the PCR. This machine would rapidly heat and cool down the sample to a series of cycles. This temperature cycling is what allows us to have DNA separated, copied, and then rebuild. With each cycle, the amount of DNA will double. So let's say we have about 30 to 40 cycles. So at the end of the entire process, we will have about maybe million to even billions of copies of the target DNA. If the virus was present in the original sample, the process will boost its genetic material and it will give us a signal to be able to pass on to the next step. Now that we have the virus, we need to detect the virus with fluorescence. This part is what comes into play, the Q in RT-qPCR. The Q stands for quantitative. This part tells us if the virus is there, but it also tells us how much of the virus is present. As the PCR process makes more and more copies of the viral DNA, a special fluorescent dye is used. This dye will create a noticeable color change. The dye will stick to the new DNA that we had previously made and make even more copies of it. The more copies there are, the brighter the sample will grow. A special machine will watch the fluorescence grow with each cycle of PCR. If the glowing signal becomes bright enough to be able to detect that, then we will consider that a positive result for COVID. But in the other hand, let's say that we have no glow or detected very little glow throughout the cycles, then the result is negative. This method is incredibly sensitive, so even small amounts of the virus can be detected. Sometimes results can even be seen before the patient even has any symptoms. Lastly, it's important to understand the results. Understanding the CT values, which tell us an important detail that we need to understand. The CT value stands for cycle threshold. This is the point during the PCR process when the fluorescence becomes strong enough to be detected by the machine. So if we have a very low CT value, this would create a sooner fluorescent appearance. But if we have a very high CT value, it means that it takes a lot of cycles in order to see any fluorescent dye. If the machine goes through all the cycles and never detects any fluorescent, then we will consider that a negative result. So CT value not only tell us whether we are positive or negative for the virus, but also tell us like a rough idea of how much virus we have inside the sample. To ensure accuracy, scientists will run controls alongside the patient sample. So the point of a control is to be able to compare and to help verify that the test is working correctly. Thanks to the safety measures that scientists use, RT-qPCR is a very reliable and important method to be able to detect COVID. So now that you know how the RT-qPCR works, think about it the next time you're taking a COVID-19 test. RT-qPCR is a very incredible tool that helps us quickly and accurately detect the COVID-19 virus in your sample. Thanks for watching, and I hope you stay safe.